Hi everyone, this is Jason, producer of Data and Biotech. Before we get started, I wanted to let you know about our latest white paper. It's a comprehensive guide to implementing machine learning models in biotech manufacturing. It's a complete overview of all the potential problems of ML adoption and, more importantly, how to solve them. To download it, simply visit connect.cordine.com/biotech-ML. We've also dropped the link in the show notes of this episode. Okay, let's get into it. Welcome to Data in Biotech, a podcast from Cordine where we explore how companies leverage data to drive innovation in life sciences. Every 2 weeks, we sit down with an expert from the world of biotechnology to understand how they are using data science to solve technical challenges, streamline operations, and further innovation in their business. This week, we sat down with Stewart Fossceco, former VP of Quality Analytics at Invitae and expert in the field of pharmaceutical manufacturing. During the interview, he and Ross discuss why experimental design and statistical simulations are valuable tools for minimizing variability in biotech manufacturing processes. They break down why stability modeling is crucial for ensuring the quality and potency of drug products throughout their shelf life, and unpack why trade-offs between stability and market demand must be considered in drug development and manufacturing. Here we go.
Stewart Fossceco, welcome to the Data and Biotech podcast.
Well, thanks, Ross. I appreciate it. Thanks for the invite.
Well, just to kick us off, could you just give us a tour through your career and an overview of how you came here today?
Sure. I actually kind of started off in grad school concentrating on molecular genetics and quantitative genetics and combining those things, and got into statistics, of course. And then through my experience at Virginia Tech, where I received my graduate degrees, I kind of found a little niche where applying statistics to biological processes and research, really enjoyed it and found that I could really help people, and drove me down to a whole career path over the past 30, 35 years.
Over the course of that career, what can you share some of the high-level considerations that you've seen when working in biotech manufacturing and specifically around quality improvement strategies or QA?
Over the course of the last 25, 30 years, what we've really kind of seen is the pharmaceutical and biotech world embracing things like quality by design, where quality of a product and robustness of that product and the processes that produce it are developed, and that begins its early stages much earlier in development as opposed to later, once it's sort of been submitted, accepted, and then manufactured. I believe that in large part, that's a response to the recognition that we have levers that we can pull during development that allow us to create a product and manufacture it to the highest quality standards, and do so as effectively and efficiently and cost effectively as possible, and minimizing that overhead and costs associated with it, with failures as they arise in that manufacturing process, which can be very costly, of course, for both the pharmaceutical, biotech companies, as well as the patient. You want to keep those products as price efficient as possible for them.
Yeah. So what does that mean in terms of being involved in sort of the early development of these drugs and bringing them to market? What are the kinds of things that you bring to the table when you're there in the early parts of that process?
Yeah, in the early parts of the development process, what you're really looking for are to identify those factors that are driving the quality of the product, particularly around areas or other aspects of the process that you may not have as much control over. The weather, for example, where you may be manufacturing. So if something ends up being very temperature sensitive or light sensitive for that matter, you can pull various levers during the manufacturing process to then make that entire process much more robust to changes in temperature or light conditions, humidity, of course, if you're working with various things that are maybe like a dry tablet formulation. There's just a plethora of information and research over decades that span not just the pharmaceutical and biotech industries, but also the car manufacturing and other manufacturing industries that have looked at how to design through experimental design where we identify those factors and then could begin to utilize those to develop either the formulations and or process itself in combination with the formulations that end up going into the patient to create that kind of response for everyone.
So I'm imagining, like, as you're bringing a drug to market, there's sort of a list making exercise or a brainstorming exercise where you're sort of writing down all of the factors that need to be controlled for or need to be varied, and then sort of designing an experimental regime around those factors to understand, how this therapeutic treatment is going to respond to variations in these different factors. Am I thinking about that right? Or can you walk through the process of like how that looks?
Yeah, absolutely. And it even starts even one step earlier than the factors themselves and designing those experiments, in that when we think about the measurement systems that we have to measure the qualities that we're interested in, it could be efficacy or potency, maybe a particular drug product degradant that forms over time, which comes into play in our stability programs later on. The actual measurement systems themselves, we have to be sure that we understand the variability in those. Because if the variability of that measurement system exceeds what we can tolerate in the process itself, then we'll have to work on the actual measurement system to bring down that variability so that we can measure it very precisely. And then once again, we get into the world of experimental design where we're looking to make the assay process. So again, we're back into this mindset of we have another process that we have to optimize essentially. And we want to optimize that assay process and identify those factors that make it robust to various conditions in which we will operate it, so that whenever we get a result, we know that we can trust that result and we can minimize that variability, because then that plays into the kinds of decisions that we have to make about the product. And not just in manufacturing, but that even goes all the way into the clinical trial realm, where if there's too much noise in the product, that could impact our decisions in the clinical trial process, where those assays might also get used.
How do you go about optimizing or minimizing the variability of those assays so that you can be more confident in the clinical results?
So there may be steps along the way that you develop the assay. You kind of have various different components chemically that you're using, say A, B, and C different components. And those get used, and then you may have various incubation times that you have during that assay process. And there may be several steps where you have different incubation times and possibly different temperatures, let's say. So you have incubation time, temperature, and the actual components that are part of that assay. So you look at designing levels, say, all right, we want to test a high, low level, and maybe some intermediate levels within this process, high temperature, low temperature. Again, if we're running the assay in the middle of the winter and our lab happens to be cooler and or less humid than in the summer, and it's maybe a little less temperature controlled environment, how will that impact the final outcome? Because where we could end up is, and what we want to do is we want to define a space where we can operate that assay and then come out with a reliable outcome. And we can oftentimes use a combination of experimental design and then statistical simulations to demonstrate how those factors are impacting the assay and how some of the activities that we do during the execution of the assay, where particular thing that may make sense to us intuitively actually hurts us. So for example, let's say we are assaying values that are expected standard concentrations. And we're going to look at 240 of those at a time. They've been created under a process that's in control and with an expected concentration. And those are going to all get combined into an assay and then used during that process and evaluated. We create the solution with all 240. We measured those beforehand. So we know we have an estimated concentration of each one of those. It goes into our solution. So on average, we have a very good estimate of the average concentration of each of the 240 component mixture. Intuitively, we think as scientists, we should measure that one more time once we put everything together. So we measure it. We take a sample from our solution we just made, and we measure it one time. Now what's happened is we've taken all the assay variability that we reduced by the square root of 240, because we just took a sample of 240 times, we put them into solution. It's an average of 240, which we sampled and knew. We have an estimate for each one of those. Assuming it follows, say, a normal distribution, kind of normal variability, thing behaving well, we have a pretty good estimate. The estimate of the standard deviation of that mean solution is this variability divided by the sample size, and the standard deviations, the sigma divided by the square root of n. So we have a pretty precise estimate. Intuitively, though, we think, oh, let's just be sure. We're cautious people. So we just want to check it one more time. So we sample that whole combined solution and we measure it. We've now taken all the variability of the assay that we reduced by that square root of 240, and we've interjected that variability, all of it, back into the assay. And so that's where intuitively,
because your sample size is 1. Am I thinking about that right?
Size is 1.
Yeah,
exactly.
Okay.
Yeah. So it's a very interesting thing where you can actually see it and you can simulate that process and show it's like, oh my gosh, we should just run with what we've got. As long as our other upstream process is running and in control and it's behaving as expected. So that's one example. Another one is, let's say we have an assay and it's for a product that we know well, we produce, and it's very well behaved. And that assay has been worked on for years, let's say. And the variability is as low as we're really going to get it unless we want to completely change the process, which would involve additional submissions to regulatory agencies, say globally. It's a lot of work and a lot of cost, and cost prohibitive, in fact. And let's say we use this assay for drug release or a component release for a diagnostic or something of that nature. We know that assay variability. We know it well. We know our process variability very well. And we begin to examine those ratios. At some point, when the assay variability begins to exceed a certain limit, what happens is if you test your material at release and you make a decision, you could be making that decision based on just random noise and nothing else. You would be better off not touching it and going ahead and saying, all right, that is our best estimate due to the assay variability. And what you want to do then is, what are the limits within your specifications? What are the limits within that space that you say, okay, if we're outside of these limits, that's going to be outside of what we would have expected at random based on our assay. So we think that we did have a bit of a shift. So in, say, concentration of the product prior to release. So let's adjust that concentration of the product either up or down by adding more concentrate or diluting it a bit, and then go ahead, test it, make sure it's good, and you release it at that point. So that it comes down to, like, where are you in those processes, and understanding of that assay variability and how it can impact your business decisions later on.
There's kind of an intuitive nature where, you know, if you're of a scientific mind, you always have this belief that, like, experimenting more is always better.
Yeah.
But there's also sort of the timing aspect of where in the process you should be sampling so that you can get sufficient signal on both the variability of from the assay, you know, based on its variability on the drug that you're bringing to market, so that you can make intelligent decisions. But if you're experimenting at various points in the process with different sample sizes, then you can get conflicting information that is challenging for both decision-making processes, but also has, I'm assuming, regulatory implications as well, because because the assays that you're using and the way that you're sampling and the design of experiments that you're using is subject to that sort of regulatory scrutiny as well. Am I thinking about that right?
Absolutely. In that last example I just used, that's a good example of where you have a manufacturing process set up. And in that manufacturing process, you lay out what these decision points are. And because you know the assay variability and the process variability, you can say, okay, well, at this point in the process, we'll take that measurement, and we know that because our process, and here's a key point that I think is important to understand and keep in mind, is the assumption there is that the process making that product is in control and is centered where you say it's centered and where it's estimated to be. Beyond the deviation, beyond that centered part of the process, when you get outside of that, that's where you say, oh, okay, we didn't dilute enough, or maybe the concentrate was a bit more concentrated than we thought, and so it's a little too concentrated, so we need to dilute a little bit more. So it really becomes part of your manufacturing process. And that's where, from a regulatory standpoint, they will come in and say, okay, good. You have a process that's in control and you're able to measure, what your variability is of both your assay, your process, and you're simply not going to fiddle with it if it's within this space, because on average, you're probably, if you tested it again, which you don't want to test into compliance, but if you were, say, do the thought experiment and execute that, you test again, if you're inside those bands, you're more likely to be closer to the mean because of the assay variability.
Yeah, that makes a lot of sense. So, you mentioned sort of statistical simulations, like playing a role in both constructing the assay and understanding how the assay is performing. What are the sorts of simulations that you use in a situation like this? Is this like a Monte Carlo simulation where you're just sort of like changing variables and doing random selection, or like how does that work?
Yeah, I think it's an incredible tool that we have. And because it takes probably a bit more time than fitting like maybe just a quick regression line, sometimes a little bit underutilized, really. But it's a great tool. The simulation, Monte Carlo simulations and so forth, are a great way to take a process, map it out, and then each step along the way, oftentimes you'll have data and measurements that you can use to then inform, okay, here's how much variability is being interjected into that process at that step. And you kind of step through it, because the question will be is like, how is it transferring itself through the rest of the process? That a small change early in the process could get magnified and really impact it later on. So maybe that's where you want to spend a lot of time to really gain control of your assay, or do something to drive down that variability early on, as it has magnified impacts later on in the process. So it's very helpful there. It's very helpful in so many places. Of course, used a great deal in clinical trials, as well as when we talk about efforts in looking at stability modeling and how that impacts cold chain and supply chain types of decisions as well.
If I'm understanding you correctly, like measuring the variability of different points of your process is really important, but also understanding sort of the dependencies between those variabilities and which points in the entire process as a whole is sort of very sensitive to is one of the insights that these models tend to surface. And so it helps you to focus your efforts in terms of where you want to concentrate on minimizing that variability.
Absolutely. What I've always found interesting is sometimes the outcome of those are, again, kind of counterintuitive, because scientifically we're kind of trained to like, oh, well, let's measure that one time before we take action. And sometimes that, because you're taking a sample of an N01, you're actually end up hurting yourself, because you already have so many sort of steps where you've taken measures and you've reduced that overall variability of the decision point already. So it's a very helpful tool for us to use.
what I'm hearing is that there's the risk of overmeasurement, and so there needs to be a lot of upfront thought in terms of where you take these measures, when you take these measures.
As you put, I think very well, is we think that sometimes we'll take a sample of 10 or 12 or something of that nature, maybe more, whatever that sample is at a certain point. And what it turns out to be is maybe the variability of that part of the process is pretty small. It doesn't have a lot of impact on the overall decision at the end, as opposed to maybe another place where it's counterintuitive that the variability was that big and or that impactful later on in the decision-making process. So what we want to do is shift those resources to where we have the greatest impact to decrease the variability. And that oftentimes leads us back to that early development process where we're using things like experimental design and things like that to identify where those are so that we can either control it or actually put our resource, identify, well, we can't do much with this. This is a random component. Maybe we have a supplier who has a certain amount of variability in their product. And maybe we don't have a lever to pull, we find, in our experimental designs to make it robust to their changes in whatever it is their component is that they're supplying us. Hopefully we do. But if we don't, maybe what we have to do is set up some rules of engagement with our supplier where as it comes in, we'll do some additional testing and they'll have to meet certain standards in terms of how a component needs to perform before we accept it and use it.
Yeah, I think there was one last thing that sort of came up for me as I was hearing you talk was that, we sort of started out in terms of minimizing the variability of the assays and then also using those assays that have been, somewhat improved to also figure out your experimental regime that's going to help you understand the variability of the entire process and, the bounds within which you need to be. Does any of this change when you're monitoring the process, when you're at full production scale, or is it just sort of a replay of a similar thought process that you described?
The process is typically you will execute the things that we've talked about in terms of experimental designs to minimize the assay variability, say establish a robust process product. And then prior to kind of going into full-blown production, you will kind of sit down and say, okay, now we know where our variability comes from and what we can do to manage that. Now we need to validate prior to going into full-blown production that we can execute on that. So then you'll set up essentially a full-blown validation with a protocol, with acceptance criteria, oftentimes utilizing and having decision points in there to where, okay, if our end result and our decision points all fall within a certain band, we know that anything within there is simply due to random noise that we built into our process and our decisions and the quality of our product that ends up reaching the patient, that we're good. And once you pass that validation, and that's usually a fair bit of work, those sample sizes are larger. It's not a trivial exercise by any stretch of the imagination. So I don't want to underplay that at all. Once that passes, then you go into production and you can have a different type of sampling plans and strategies there for your in-process measurements and those quality measures that you're going to have. And typically those are going to be your critical quality attributes of the product. And you'll have limits on those. And those will be pretty well established. And then the sampling necessary to know that if you do see something that looks odd, that you can engage with a CAPA, some kind of corrective investigation and corrective action if one is required. It could be just a special cause in case maybe something happened. They find a lever was left on during production and things just didn't work. And it's like, well, okay, we'll put in an extra check to make sure that lever is closed from now on. So it could, I'm oversimplifying, but I wish they were that easy most of the time.
Yeah. So it sounds like the assays might be similar, but the samples that you're taking and when you're taking them might be slightly different. But basically the goal of that measurement regime is this group of products that we're producing, within the expected bounds for
Right.
the effect, the efficacy, the potency. And then when you encounter a problem, I'm assuming there's a batch that gets thrown away, and then there's the corrective regime to go in, make sure that everything looks good, and then pass it all the way through the process.
Yep. You'll have those assays that are used in development and that are developed there and characterized. Those will be the assays that will then get used during the clinical trial process on the product to characterize those clinical batches, because those will be used for relating the product properties that are important for its quality back to the clinical efficacy. And then as those get measured, those will inevitably be used along with other measures, perhaps, to release the product to the public for consumption. And again, all of that ends up being part of a large program, drugs or device stability, to ensure that throughout the shelf life of that product, that the patient will receive the dose or the quality decision that is expected. And so you are going to use that assay for many years, or whatever that process is that gets established. And it really becomes a process of continual improvement over time to really enhance and pinpoint and be able to help make the very best decisions for the patient and in real time, ultimately.
That makes sense. And I think this is a good opportunity to transition to, you mentioned stability modeling. So, we've been talking about assay variability and bringing these drugs to market. Could you give people who are not familiar with it an introduction to what stability modeling is and, why biotech manufacturers do it?
Yeah. I'll try to start with a very simple example. We have a drug product that will be consumed by the patient. And let's say the product will change over time, as most do. So there'll be some change over time. And typically, let's say that we're dealing with potency of a particular treatment. And when we first release it, we want to have it 100% potent. And then during testing and clinical trials, there are requirements for the drug to meet certain amounts of potency. Let's just say hypothetically, it's 90 to 110%. So the degradation rate or loss of potency over time becomes an important factor in the quality of that product and what the end result is and the experience and outcome for the patient. And you need sufficient amount of resistance to whatever environmental factors are going to influence that product to ensure that aspect. And companies start very early in the development process, typically modeling drug stability over time using both longer term stability programs as well as what are called accelerated stability programs, where the product, particularly if it's temperature sensitive, will be held at higher temperatures and humidity levels to model what the behavior is going to be over the lifespan, the desired lifespan of that product. And you could fit various different models to that, everything from, I'll call it simple linear regression models over time, where the samples will be taken and measurements taken at time zero or the day it's manufactured, to 1 month, 3 months, 6, 9, 12, 18, 24 months, and maybe out to 36 months for some products. Take, for example, things that are very stable, like various solid dosage forms that aren't very temperature sensitive, not very sensitive to humidity. Those can last quite a while on the shelf. Now, the other things that are refrigerated could, if they're exposed to heat, particularly high temperatures, if we think about certain geographical locations around the globe, you get into very hot environments around the equator. You have to sort of make sure the product stays refrigerated. Otherwise, you could have a pretty significant loss of potency. So that modeling process and that program becomes exactly that, a program. It becomes a stability program for every product that comes to market. And that starts very early. And we'll back all the way up to even before clinical trials, before, as that product is being discovered and worked on in discovery. And if it looks promising, There will be formulations, scientists and experts, chemists and process folks who will be working very hard on ensuring that formulation for that product or construction or engineering of that device is resistant and the best they can bring to the market. And that stability program will begin very early and looking for those optimum formulations.
So as we draw the conversation toward the end, I'm just interested in, what do you think are some of the biggest challenges that biotech manufacturers have in gathering the right data and implementing the right practices and processes to understand their assay variability, understand the stability of the drugs they're bringing to market, and, sort of optimize the processes and the feedback loops that need to be in place for that information to be in the right hands?
Some of the biggest challenges are trying to think ahead far enough about what data you're going to need to answer the questions. And then in what form do you need to make that accessible to the people who need to make those decisions? The more we can think ahead about where those decisions need to be made and who, then we can empower those people, hopefully at very early on in the process, at the scientific bench level, to make those decisions very fast with very clear statistical and quantitative assessments that give us a probability statement about how sure am I that I'm making the right decision. Again, because our intuitive selves look at things sometimes and go, I think this is the way to go. But then if I think about it, put my statistical thinking cap on, I'm like, wait a minute, let me rethink this a little bit. The data capture piece still for us becomes a barrier. So I think that's one place that if organizations really think through, okay, where are we going to make those decisions and what do we need to put in the hands of people and how do we need to structure that data to make that happen for them? I think that's
getting the data off the assay machines and into a place where it can be analyzed together with the right metadata about the different variables that are related to stability that you need, so that the sorts of statistical analysis that you're talking about can be conducted at scale and over time.
Even things as simple as the genealogy of the components that go into, say, a diagnostic test. All the different components that come in, the manufacturers, the suppliers of those components. Because if something does arise, it's like being able to tease that out analytically becomes very difficult if you don't have the data there and the metadata about all of those components. The other place I say, and I think this continues to gain steam over the course of my career, I know it has, just the philosophy of quality by design. Starting early with those experiments that we've mentioned multiple times this afternoon, where let's look at how all of these things are interacting with one another. Because in that data, in those experiments, there are going to be diamonds that we can use that are going to save us so much pain and hassle later on. And we won't get caught late, either the development process, submission, or even manufacturing process of having to figure out, it's like, now we have, let's say, so much variability in our process. We have to execute so much testing that it becomes very onerous. I think many, many industries have found that trying to test quality into a product is very difficult. It's much more productive and much more cost effective to design quality into the product and the process for robustness in the long term. A little more upfront cost, sort of pay me later or pay me now kind of thing.
Right. So, especially for small organizations with limited resources where they have sort of a biological hypothesis about how a disease gets cured, it's difficult to get that type of organization to focus on sort of like the quality attributes that will need to be in place of the therapeutic that's brought to market. And, I can imagine that asking them to spend those extra resources during that early phase is relatively difficult. So, do you have any advice for organizations like that are bringing drugs to market for how to do quality by design more cost effectively, or how to bring those considerations to bear earlier on?
Yeah, start small. You don't want to try and boil the ocean to start with. So if you start small and run even smaller experimental designs geared towards understanding how these things are piecing together a little bit. Even in the short term, what you'll probably find, or what I've always found throughout my career, is if I add up all of the small experiments that everybody did along the way, say over the course of 6 months, let's just say during that period, there were gaps that were created simply due to the way that ended up being executed because of the mindset around we have to move fast. We have to move fast. So we have to make this very first decision today so that we can get to this other one, the one we want to make 6 months from now. What I've found oftentimes occurs or has occurred is if we had paused, not too long, but just long enough to understand, okay, here's our end decision. Here's the business decision that we're really going to set our hook into, right? This is the one that's going to really matter. What are all these pieces in between? Let's just look at that. And let's design maybe 2 or 3 experiments, well-designed experiments that get us that information to make that decision with that probability statement I said. What inevitably oftentimes happened was the amount of time it took to do that, to go through the processes for everybody to understand those processes and what was needed, design the experiments, execute them, and then analyze them. It took fewer resources, less time, and less experimental runs in total than if it was done kind of in that very small sort of sequential manner.
Is the business decision that you're talking about like, submitting an IND or deciding to bring something into clinical trial or deciding to scale up manufacturing? Like what are those the sorts of business decisions you're talking about, or what are?
Yeah, it could be a business decision. At some point you're like, okay, so we need to have a clinical material made. So here's our decision point. What do we want going into this clinical trial material? And so understanding that and kind of working backwards, or maybe it could be an IND, but it doesn't have to be that far along in the process, really. It could be very much something much earlier. It could be something where you're looking at the behavior of a sustained release product for midway into, say, early preclinical testing, for example. And the preclinical decision point is going to hinge on a particular response. Well, how does what we're going to build impact the variability of how those folks are going to see that data? And once you make that link, you say, okay, here are the things we need to sort of really address. And let's not have the gaps that we might have otherwise.
Well, Stewart, thank you so much for joining us today. It was a pleasure to have you on, and look forward to connecting down the line.
Well, thank you very much, Ross. And thank you for your time. And I've really enjoyed the session.
And that's it for this episode of Data in Biotech. If you enjoyed the episode, please subscribe, rate, or leave a review in your podcast platform of choice. See you next time.